Hello, everybody. A warm welcome back to the third session of this symposium in honor of John Woolliams. I'm Chris Haley. I'm a professorial fellow at the University of Edinburgh, working both at the Roslin Institute and at the Medical Research Council Human Genetics Unit. I have been a colleague of John's through, I think, 35 years. Starting first at the Animal Breeding Research Organization, and then through various name and organizational changes over the intervening years until now, and had several privileged opportunities to work with John. My interests are largely in complex trait genetics and using various omics tools to explore those. I'm going to be chairing this session this afternoon, the privilege of chairing this session. And my co-chair is Smaragda, one of John's former students, and she'll take over when my internet or my brain goes pear-shaped. But you'll have a chance to see and hear Smaragda later on in the session. So for the first few speakers, remember that you have 15 minutes for presentation and 5 minutes for questions, and I'll. If I'm feeling like it, I'll give you a warning when your time is near up. Remember, if you're going to ask a question, that please ask it as soon as it comes to your mind. Don't wait till the end of the talk. Put it into the Q&A. Others, if you like that question, give it a like, and that means it's more likely to get asked and answered. And remember to put a name down against the name of the presenter you're asking the question of. So that if it doesn't get asked, the presenter can answer it at a later date. And remember that we will be making these talks available after the end of the meeting. So with those few points out the way, we'll introduce our first speaker, Piter Bijma. Piter is based in Wageningen University in the Netherlands, and he did his PhD in the 1990s at Wageningen, but worked closely with John and spent some time at the Roslin Institute at the time, working on predicting inbreeding in selected populations. Since then, he's done some groundbreaking work on the genetics of social interaction, and he now works on quantitative genetics and epidemiology, and we'll be talking around this subject today. So over to you, Piter.
Do you see my screen now?
Yes, we do see your screen.
Okay, good. And I guess I'm live now.
You are, yes.
Yes. Okay. So I first want to thank John and the organizers for inviting me to speak at this farewell of John. I think it's really an honor to be invited here. So this afternoon I will show that indirect genetic effects, that they contribute to the majority of the heritable variation in the prevalence of endemic infectious diseases. And here you can see my two co-workers. But before going into infectious diseases, I will say a little bit about my history with John. So John was my principal supervisor during my PhD, even though I was based in Wageningen. And in those days, this was still at the old Roslin Institute, which is actually very different from the new Roslin Institute. And in these days, John was working on some of his most important work, I think, on the long-term genetic contributions. So this was an excellent time actually to visit John for a PhD that focused on the prediction of the rates of inbreeding. And I particularly remember that John was a very critical thinker who really wanted to understand things. So we should not be satisfied with say methods that work, for example, but we really have to understand why they work. And I appreciated that a lot. But John was not only a good scientist. I mean, John is also a very sweet person, and I want to mention one anecdote. I visited Scotland, or I visited John essentially for a few days, and then he invited me to go hiking around Peebles. But I was there only for a few days, so I didn't bring hiking shoes. And John was ready with carrying his boots on. And then when we were hiking, I noticed that he no longer had his boots on, but he had his decent shoes. So he changed to his decent shoes simply because I forgot my hiking shoes. So that was very sweet, John. That wasn't really needed, but it was very sweet. So after my PhD, we collaborated again, focusing on infectious diseases in the project of Debbie. And there the focus was on say, hidden genetic variation that may be present in the form of genetic variation in infectivity. And that brings us to my topic of today, which is on infectious diseases. And I will look into the prevalence of an endemic infectious disease. So it's not an epidemic disease, but an endemic disease that is present, say, like mastitis, but for example, not the COVID. So the trait that I will be looking at is a binary disease status. So where non-infected individuals have disease status 0 and infected individuals have disease status 1. And I will also be looking at the prevalence in the population. So by definition, the prevalence is the fraction of individuals that is infected. So the prevalence is between 0 and 1. But obviously, the prevalence is simply the average of individual disease status, because the infected individuals have a 1 and the others have 0. So this means that response to selection in prevalence is the same as response to selection in disease status. So we are looking into binary traits here, and that has a bit of a history in Edinburgh. Here you see a picture of Alan Robertson, and in a landmark paper by Dempster and Lerner, Alan Robertson showed that binary traits have a maximum heritability at a prevalence of 1/2, and that heritability goes to 0 if the prevalence goes to 0. So this means that heritability vanishes if the prevalence goes to 0, and that suggests that selection cannot eradicate a disease. However, if we think of infectious diseases, a key property, of course, of infectious diseases is that they can be transmitted, and that completely changes the picture. So if we take an epidemiological perspective, then the prevalence of an infectious disease, it's like an emergent trait of the population. It's not a property of an individual or an average of individual traits, but it's a property of the population, just like, for example, the size of a termite colony that you might see in the wild. So we have to take a different approach. So if we take an epidemiological perspective, then it follows that the prevalence is determined by the basic reproduction number, which is known as R0. Here you see the equation, and in the figure you see the relationship depicted. So for R0 smaller than 1, there is no disease. And if R0 increases, we see the prevalence going up, initially steep, and later on the relationship becomes flatter. Just for the sake of completeness, what is R0? R0 is the basic reproduction number. So it's the number of individuals that gets infected by a single infected individual if the rest of the population is non-infected. So essentially in the beginning of an endemic or an epidemic. So now that we know from epidemiology that R0 determines the prevalence, then for breeders, of course, the next question is, what are the genetic factors that determine R0? Well, and we have already seen something about that in today's seminar. So obviously there is the population mean contact rate, but that's a fixed effect for the entire population. But the genetic effects are the susceptibility of individuals to become infected, the infectivity of individuals, so that's their propensity to infect other individuals, and the duration of the infectious period, which has to do with the recovery. So together, these three genetic factors determine the genetics of R0. So the previous slides, they sort of provide some background for my talk. I will now talk about the focus of this talk. So the focus of this talk is to look into the difference between response to selection in prevalence and the breeding value for individual disease status. I mean, we know that prevalence is just the average of individual disease status, but I will show that the breeding values are completely different. So I will take an approach based on the breeding value for R0. And I will look only at genetic variation in susceptibility. So there are no additional sources of genetic variation, such as infectivity, superspreaders, etc. This is just a genetic variation that we know exists. And I will look at reasonable values, so say a heritability of binary disease status of only 5%. And importantly, I will assume that the pathogen replicates only in the host. So there is no factor, no badges, for example, like with TB. And my aim is to understand the response to selection and the role of indirect genetic effects in response to selection. Well, obviously there are some methods, but I don't have the time to go into methods. But just to briefly summarize, essentially the only thing I do is to integrate some quantitative genetics in standard epidemiological theory. Well, and if we do that, it turns out that an obvious approach is to define an additive breeding value for the logarithm of R0. And if we have done that, working from that onwards, we can find the ordinary breeding value for the Z status, so which turns out to be a function of the breeding value for R0. And that's given by this relationship here. So it's a simple, P is the prevalence. So the breeding value for individual disease status is a simple function of its breeding value for R0. And the interpretation is just as an ordinary breeding value. Suppose we have a population with a prevalence of 20%. We have a breeding value of -2%. So then we expect that 18% of the time this individual has disease status 1 and the rest of the time is non-infected. So this is just a prediction. The breeding value here is just a prediction of the individual phenotype. Again, starting from this breeding value for the logarithm of R0, we can also find a breeding value for prevalence. And this breeding value refers to response to selection. So in that sense, it's like a total breeding value. So the interpretation of that breeding value is that response to selection is equal to the change in the average breeding value for prevalence. So again, suppose we have a population with a prevalence of 20%. We select a bunch of parents that on average have a breeding value of -2%. Then the prevalence we find in the offspring on average is 18%. So the breeding value for prevalence is predictive of response to selection. However, if you now look at those two breeding values, then you see that they differ by a factor equal to the prevalence. Here you see P, which is not here. So the breeding value for ordinary disease status is a factor prevalence smaller than the breeding value for prevalence. So what does this mean? Well, let's look at an example to illustrate this. Suppose we have a population with a prevalence of 20%, and we have an individual that has a breeding value of -2%. So for this individual, we expect that 18% of the time it has disease status 1, it is infected, and the rest of the time it's non-infected. However, if we would select such individuals, so individuals that have a breeding value for own disease status of -2%, if we would select such individuals as parents of the next generation, then we get a factor equal to 1 divided by the prevalence more. So we had a prevalence of 20%. The breeding value was -2%. We have to upscale that by 1 divided by the prevalence, and that means that prevalence goes down to 10%. So we had a prevalence of 20%, a breeding value of 2%, but because prevalence was 20%, we have to upscale by a factor 5. So response to selection is 5 times greater than we expect based on our ordinary breeding values for disease status. Well, and that changes the response to selection quite a bit compared to current belief. So here on this slide, I look at the heritability of binary disease status, and that's very similar to the result of Robertson. So the heritability for binary disease status has a maximum at a half and moves to 0 at a prevalence of 0. The values are a bit different, but the pattern is the same. But if we look at the ratio of, say, the genetic variation that we can use for response over the phenotypic variation, so if we look at the genetic variation that matters for response to selection, then the pattern is completely different. We see a strong increase in the genetic variation that we can use for response to selection. And these are inputs for different magnitudes of genetic variation. The value of 0.5 corresponds to an observed scale heritability of something like 5%. So we are not looking at very high heritabilities here. So these are realistic numbers. So based on this, we expect that we can get more response to selection than we actually currently believe. So we of course tried and look into that. So from classical breeding theory, we expect that response to selection is the product of the intensity, the accuracy, and additive genetic standard deviation. And what I did here, I take this product and I use the ordinary values that we find for binary disease status. So these are the values that we know, and then I upscale this predicted response by a factor 1 divided by the prevalence. For example, if we have a prevalence of 20%, I would have to upscale by a factor of 5. And I compare this prediction with stochastic simulation of endemics, simply following a standard theory and applying mass selection. So there is no theoretical prediction involved here. And if you look at this figure, so on the x-axis is the generation, on the y-axis are the prevalences. I have two populations, one starting at a prevalence of 95% and the other starting at a prevalence of 50%. The small circles are the actual prevalence observed in the simulations. The blue line is this prediction, where I upscale by factor 1 over p. And the dashed red line is what we would expect based on classical breeding values. And then we see that this upscaled prediction here, the blue line, it matches very closely the simulations, both for this population starting high and the one starting at a half. We also see that the classical expectation is way off. Response is much smaller than we observe. We see that the response is actually quite large, particularly in the final generations where the prevalence is low, response is very large, and the disease goes extinct in the final generation. So why does this happen? Well, I think here we have to give some credit to Steve Bishop, who has been talking about this topic time and again. The reason that this happens has to do with feedback. Say we have susceptible individuals, they can become infected and they recover again. And the transmission of susceptible to infected depends on the number of infected individuals. So there is an indirect effect. If we reduce the number of infected individuals, then also this transmission rate goes down and we get more response. And that effect is way stronger if we are at low levels of prevalence. Here you see the relationship between R0 and prevalence, and that relationship is much steeper at low prevalence. And that's why we see such a strong acceleration of response if we come at lower levels of the prevalence. So my conclusions are that most of the genetic variation in prevalence is hidden in indirect genetic effects. For recovery, which I didn't show, but for recovery, everything is exactly the same as for susceptibility. And for those two traits, a fraction 1 minus the prevalence is hidden, which is usually more than 50%. And for infectivity, actually all genetic variation is hidden. So this means that there is either way more host genetic variation for response to selection than we currently believe, or there is some other huge factor that we are still overlooking. But this suggests we can get much more response than we currently think. A key point here is that I looked at a pathogen that does not replicate outside the host. Finally, I want to come back to John. I mean, John, I want to thank you for an excellent training. I really learned a lot. I also really appreciated your dedication and support, both for my PhD thesis and later on in the work of Debbie. So here we see, I think we see a typical picture of John, maybe, well, very dedicated, maybe slightly more concerned than would be optimal for his own well-being, but really dedicated to solve problems. So this concludes my my presentation. Thank you.
Thank you, Peter. That was really intriguing. Can I just, so you mentioned that this is due to not appropriate for pathogens that have an alternative host. And that is essentially, is that because effectively there are many more indirect effects you're not accounting for there?
Well, if there is say a factor, then the pathogen can also replicate in the factor. So they say that the benefit of reducing susceptibility is smaller because the pathogen has another route to multiply. I mean, this strong effect occurs because if you reduce susceptibility, then the pathogen infects fewer individuals, which in turn reduces infection pressure. But if the pathogen, for example, can replicate in the soil or whatever, then you don't have this strong feedback.
Okay, so it implies that it's direct individual to individual infection.
No, not necessarily. I mean, it's completely fine if the pathogen can survive in the environment.
Okay.
But it should not increase in numbers in the environment.
Right. All right. Okay. Good. Thank you. So a couple of questions in the Q&A from Andres Lagara. Is there any implicit assumption of a single herd such that any individual can transmit disease to any other? What happens if you consider separate herds?
Well, essentially, what I mean, we look here into in a way in a basic epidemiological model where the contexts are random. So this would apply within a herd and also within another herd. I have not been thinking about transmission between herds. I mean, what we see if in the final generation of response to selection, then the disease went extinct. That's essentially because of herd immunity. So if you would then bring a new infected individual into a disease-free herd, that herd would again become disease-free.
Yeah. Okay. One more from, well, there's a couple more questions, but let's ask one from Andrea as privileged moderator. Brilliant work, Peter. Can you maybe use published results on IPM response to selection to estimate these effects?
I don't know, but we can talk about that and look into that. Yeah, I'll come to you to discuss this, Andrea, because that would actually be quite neat if we could. Yeah.
I guess in that case, because it's a single gene, basically the heritability changes as you select. It's more than just a simple quantitative trait.
Yes, but the strong feedback say that the factor equal to the prevalence difference between that doesn't depend on the genetic model.
Okay.
So that could work actually.
Right. Good. Okay. Thank you very much. There are several more questions. If you could look to answering those later on, that'd be great. That's a wonderful talk. Thank you very much, Peter. Now, do we have our next speaker in the room?
Unfortunately, Steve is not here with us, but we have Joanna as next speaker.
Okay, so we're going to hand over to Joanna and hope Steve turns up. So Joanna is one of John's many former students, and she looked at in her PhD with John into genomic prediction in chickens. She subsequently worked at Roslin, but she's recently moved to the Kennel Club. She did work at Roslin on some genetics of health and behavior in dogs, and she'll be talking today about some of the dog work. So over to you, Joanna.
Thank you.
Joanna, please share your screen.
Right. I think that's the wrong presentation. Can you see the presentation?
It's okay. It's dogs.
Well, I stupidly opened Steve's presentation as well, so apologies. You'll have to give me just 1 second.
Nothing is visible still.
Right. Oh, this is a nightmare. Apologies for this. Don't seem to be able to close Steve's presentation now. which is a little worrying. Oh, dear me. Right. Apologies, it will take just one more second. So just to give a bit of an explanation, I am preparing to give 2 presentations today. One of them is completely ad hoc and it's not mine. That's why I've had a little bit of an issue with the PowerPoint, getting confused which presentation to show. But hopefully now we should be on the right track. Right. There we go. Can you see it now?
Yes, we will see it. It's live. You can go.
Okay, great. Thank you very much for the introduction and apologies for the delay. I'm deeply honored to be presenting in such a great crowd of people. And particularly, I'm honored to be presenting in John's symposium. John has been pretty much a constant in my scientific career. So as you have heard, he was my PhD supervisor, and recently I was one of the last 2 postdocs that he left behind when he retired. I try not to think too much into the fact that I was his last PhD student and his last postdoc, which may mean that I was the last straw that pushed him over the edge. One of the things that I always really appreciated in John is that even obviously he's a great authority in terms of statistical genetics. He also is always interested about how this theory can be translated into practical breeding of animals and plants. And so in my talk, I would like to change the gear a little bit and move to the complexities of the practical applications of management programs in the example of pedigree dogs. In my presentation, I'll first give you a very short overview of what we know about inbreeding in UK pedigree dogs. Then I will I will tell you a little bit about why management of inbreeding is such a complicated subject. And then I'll give you just a few results from the preliminary analysis we have done on a Mate Select, which is an online tool that I'll describe later in detail. So let's start from the genetic diversity in pedigree dogs in the UK. So of course, pedigree dogs are close populations. They have been under varying degrees of selection, so it's only inevitable that we're going to see some degree of inbreeding. But because of the special place that dogs play in our society, the inbreeding in dogs is a hot topic, which is gaining quite a lot of controversy. So what do we actually know about inbreeding in dogs? A few years ago, Tom Lewis, my predecessor at the Kennel Club, and also John's PhD student, published a paper which looked at genetic diversity of all pedigree dogs in the UK. And he found that although we did see very high rates of inbreeding in the 80s and 90s, over the last 2 decades, the trend is actually quite favorable. We see the reduction of the rate of inbreeding, and in certain breeds, we're actually seeing some modest restoration of the genetic diversity. This trend in rates of inbreeding seems to be unrelated to the breed census size, with the small numerically breeds, which are presented here on the plot on the left side, having more favorable trends than the common breeds, the popular breeds, where you would expect that the numbers should be high enough for selection of the individuals that will minimize the inbreeding. Unfortunately, in pretty much all the breeds, we have seen the evidence of popular sires, and of course we know that popular sires are a significant driver of the rates of inbreeding. And further, even though we see some positive trends in the rate of inbreeding, the effective population size in some breeds is a cause for concern, with 68 of breeds, sorry, of 116 breeds having NE estimated below 100, and 29 of those having an E below 50. So obviously, this situation is not really acceptable. We need to do something to improve the genetic diversity in many of the breeds. That's why Tom has been having a lot of discussions with John about how we can improve things. And one of the ideas that they had was using long-term contributions in the management of inbreeding. I don't have time to go through all the results that they found, but the one that I would like you to remember for the rest of the presentation is that long-term genetic contributions in dogs can be made within the lifetime of a dog, but there is a variation between breeds and of course sexes. And at the best case scenario for males in a pointer breed, we can predict the long-term genetic contributions at 4 years of age of a sire. So now let's move to the social side of things. Why those management programs are so difficult in dogs? Starting from the breeding structure of dog populations. When we look at the breeding structure of livestock populations, such as pigs and chickens, we usually have a nucleus, multipliers, and final population, with majority of the selection carried out in the nucleus, which is usually a very small proportion of the total population. Because the gene flow is pretty much one-directional, if we are quite smart about how we handle the nucleus, we can, in effect, control the whole population by applying schemes just to the nucleus. When we compare that to the dog breeding populations, in theory, we could portray them in a similar way, with the champions, either show or work champions, at the top of our pyramid, followed by the general breeding population, and then at the bottom having pets. I've put pets here in the quotation marks because obviously all dogs should be kept as pets. But in the dog world, pets are defined as individuals that are not intended for breeding at the time of purchase. So in theory, we have this nice pyramidal structure, but unfortunately, the gene flow is very unstructured. We've had many a champion bred from pets, and at the same time, we've had many champions that never bred animals that went into the breeding pool. So that means that any sort of management system that we would like to apply will in fact have to encompass the whole population with the flexibility of moving breeding animals in and out of our system. Secondly, the system has to be really flexible in terms of the numbers of individuals that we have within a breed. So in a normal situation, we have breeds hugely varying in sizes. We have some new breeds that will have less than 1 dog registered per year, to common breeds like Labrador Retrievers, for which we've had over 35,000 puppies registered for decades now. On the other hand, within the same breed, we can see very drastic changes in numbers. And a poster boy, of course, here would be the French Bulldog, which over the recent years has seen pretty much exponential growth. So we've gone from about 200 puppies registered in 2000 to 37,000 French Bulldog puppies registered in 2018. So obviously that will require the system to be fairly dynamic. Another complexity comes from the fact that the breeding decisions are made by many independent and small-scale stakeholders. In a study done on the French pedigree dog breeders, nearly 70% of them describe themselves as hobby breeders, producing 1 litter a year or less. And this is very similar in the UK, with some unofficial figures showing that the number of hobby breeders might be actually even larger. Now, if we think about applying systems that will take into account selection goals as well, we have very variable selection goals, not only between breeds, but also within the breed, and also varying intensity. So in certain breeds, those selection goals are so different that we actually have subpopulations that don't really mix. And those subpopulations could be sporting dogs, working dogs, or show dogs. But even within the same subpopulations, for example, show dogs, keeping everything else the same, some breeders will concentrate on the angulation, other breeders will concentrate on the head shape. So it would be extremely hard to clearly define a single goal that we would include in the system. Now, competing interests, that's a controversial one. Obviously, competition exists in all walks of life. However, in dog breeding, this can be a leading cause in the decision-making process. Some breeders breed dogs just so they can beat another breeder in the show ring. And of course, that will have an impact on how willing they are to collaborate together to make breeding decisions. This leads us to very complex motivators behind breeders' choices, and there is just too many of them to mention here. But one that we can compare to livestock and plant breeding is the economic performance, because it's easily quantifiable. Unfortunately, in dog breeding, economic performance is frequently quoted as having low priority, and very often it's even frowned upon. Dog breeders will actually get offended if you suggest that they should breed for money. On the other side of the coin, we have the target customer, our puppy buyer, and unfortunately that group is equally complex. You'll be glad to know that I'm not going to discuss them too much. But even if we stick just to the economic side of things, you will have people that will want to get a puppy as cheap as possible when confronted and ask whether they would like to pay more for a puppy that has a good pedigree, let's call it. They will say that they don't care, they just want a pet. On the other hand, we will have people that will pay in excess of £10,000 per puppy. That is usually very common in fashionable breeds of more unusual colors. So no help from that side. And on top of that, we have breed differences. We have very strong breed cultures that affect how breeders collaborate together or don't. And also that impact on the willingness with which they accept monitoring and management schemes. So hopefully you have a slightly better understanding now why the subject of managing and breeding in dogs is so complex. But the good news is that it's not a topic that is being shied away from. In 2011, the Kennel Club in the UK, together with Animal Health Trust, released an online tool called Mate Select, which is available to pretty much anyone. You can put the name of a dog and you can get statistics for the coefficient of breeding of the individual prospective mating breed, and health test results were available. So this is what the outcome of Mate Select looks like. On the left-hand side, we have the individual result. So we put the name of the dog, Milky Brandy Biscuit, and we get his coefficient, inbreeding coefficient, compared to the breed average. If we consider this dog for breeding, we can check the inbreeding coefficient for the puppies that would originate from that litter. And in this particular case, because we have an example of a close mating, we get a warning from the Kennel Club that those puppies would not be registered. As we've had before in PAM stock, since 2014, Mate Select has been expanded to include EBVs for hip and elbow dysplasia. And the Kennel Club's plan is to have it available for all breeds for which there is enough phenotypic records to derive reliable EBVs. Right now, it's about 30 breeds for hips and a few less for elbow dysplasia. So Mate Select was one of the first tools of its kind. There is a couple of others worldwide that have similar function, but nobody has ever looked at how this data is actually being used. So about a couple of years ago, Tom Lewis, John Woolliams, and I applied to Kennel Club Charitable Trust for a grant money so that we could actually look at the usage of Mate Select and combine that with the information that we can get from the pedigree data. We wanted to learn as much as we could from the breeder's behavior and assess how the tool is currently doing and what we could do to develop it further. Specifically, we were thinking about changing the inbreeding coefficients into things like contributions, for example. We were joined by Mateja Janisz, who's our producer for today. And in the next few slides, I will show you a couple of preliminary results that we've had from this project. First, we looked at how popular mate select is within the UK and also across the world, and we found that it is used by breeders pretty much worldwide, with the main concentrations obviously in the UK and in Europe. We are very happy to see that the popularity of mate select remained relatively constant, and interestingly, we see cyclicity in the number of searches, with the peak in the spring and another smaller peak in the autumn. Then we looked at how popular mate select is in breeds of different sizes, and we found that the heaviest usage was noted for breeds for which we had 70 dams registered within the studied period, and density of searches decreased later on with the increasing population size, dam size. Of course, we do see variation between breeds of comparable size. And an interesting result here is that the breeds for which we have the EBVs available have higher density of searches, but it's not really related to availability of the EBVs in the system, because those breeds would have a higher frequency of searches even before the EBVs were added to their own breed profiles. So, when we looked at the usage of the data, one of the main questions that we were asking ourselves was how many of the litters that are actually born on the ground have been searched for prior to mating. So we've calculated that statistic for the dams in our data, and although there was an indication of the breed differences, in majority of the cases, unfortunately, the litters that are born are not searched for before they are produced. Then we had a look at the time frames if we wanted to apply some advisory thresholds on the usage of both dams and sires, what would be our time limits. First, we look at the sire's age at litter, and again there is quite an obvious difference between breeds. Labrador Retriever is probably performing the best with a fairly nice distribution, and other breeds we see that majority of the production of a male happens before he reaches the 4 years of age, which is the age at which we are hoping to be able to predict the long-term contributions. Moving on to the dams, we see a similar picture. The productive life is fairly short for the dams. This is partly biological reasons, partly limitations applied by the Kennel Club. The dam cannot be younger than 1 year old at the time of mating, and she can be older than 8 years old at the time of birth of the litter. But without a doubt, we do see a skewness towards dams that are single producers, so they only have one litter in their lifetime. So, message that I would like you to take from this presentation is that management of inbreeding is definitely, management of inbreeding in dogs is definitely a topic of high priority, but there are many sociological aspects that we need to first tackle. Mate select and other tools are popular. They are used by breeders and owners, but it appears that the breeding decisions are made independent of it. When it comes to the practical application of advisory thresholds, we know that we will have a very, very short window in which we will have to apply those thresholds. So with this in mind, we are hoping that given the unique data set that we have, we will be able to come up with a system that will account for the differences in breeds, for the management of the genetic variability in pedigree dogs. So I would like to thank my collaborators whose work I shamelessly stole for this presentation. And of course, more than anything, I'd like to thank John for being there throughout my scientific career and supporting me in following my dream. Thank you so much. I'll be happy to answer questions.
Thank you, Joanna. That was great. Thank you very much. There's actually a number of questions in the Q&A. I'll skip over one from Lutz, who's asking about how many dog breeds there are in the world and how big and how small they are. He'll probably tell you that You can breed mice more different than that. So someone comments it's a task of teaching the public on what inbreeding is and the consequence of dogs. Of course, I thought when you said that if you told someone, asked them if they wanted to have a good pedigree, they said they just wanted a pet. But probably if you told them that actually, do you want to have a healthy dog, they'd say they wanted a healthy dog. Is that not right? Is it just a question of giving them the right message?
Well, the problem is that you will get dogs that have fairly high inbreeding coefficients and they don't seem to have an obvious health problem. So for the survival of the breed as a population, of course, these individuals are not the best choices, but for the puppy buyer that gets the dog, they might actually be quite happy with what they got. So there are very different priorities for a puppy buyer and a breeder, and a breeder with genetic background. Yeah, I hope that this answered your question. Yes. There is definitely a lot of miseducation about inbreeding in dogs, because this is such a controversial topic. There is a lot of some people sometimes will grasp a little bit of knowledge here and there, and then they will misinterpret what it actually means. So quite often you might get across a person that will import a dog into a numerically small breed, and that dog, of course, is unrelated to any dog in the population, so they will advertise that they are working on increasing the genetic diversity of the breed within the population, but then they will go on and use that sire on every single breeding done in that breed in the country. So there is definitely a lot of scope for the education of breeders, and we do have to take into account that dog breeders are a very variable group. So what I mentioned about most breeders being hobby breeders, some of them will be hobby breeders with background in biology and genetics, in medicine, and they will approach things differently than hobby breeders that actually have no interest in dogs other than having a dog on their couch, and they like puppies, so they will approach their neighbor who has a dog of the same breed so they can produce puppies. So we have two very different, well, anything in between those two extremes on the scale of education when it comes to dog breeding.
Yeah, and some are maybe not the ones breeding for show, but some of those breeding to sell have done very well over the past couple of years, because, or last year at least, as the price of puppies has gone up 3 or 4 times during the pandemic. Yes,
that's absolutely true. Having said that, an interesting thing is that majority of those crazy prices that people are paying, they are paying for dogs that are actually not registered within the Kennel Club. So I did mention about the French Bulldog's popularity, and the most extraordinary prices that we have observed for French Bulldogs are actually for dogs that are not of registered colors. So they will not be registered in the Kennel Club's database if they have a color that is allowed in the breed. So
okay, well, thank you very much, Joanna, for an interesting talk. We have to move on now, and I think we now have Steve Lee in the room, and so we'll move to Steve. Steve has worked on the genetics of trees and their improvement at Forest Research, which is an institute just down the road from the Roslin Institute. I believe you're retired now, aren't you, Steve? But anyway, he worked with John, actually worked with John for almost 20 years, I think, from a period of when they were looking at variance component estimation through to the exploitation of genomic data. And Steve is talking to the intriguing title of About Breeding Time, or maybe it should be About Breeding Time. Anyway, I'll leave that to Steve to introduce, and over to you, Steve. Welcome.
Steve, please unmute yourself and share your screen.
And share my screen. Right, can you see and hear me now?
I still cannot see your screen.
You can't?
No.
What's wrong there then?
There should be a button below the screens, so somewhere in between us, below your screen, there is a button to share. There you go.
Okay, right. Technical difficulties. Can you hear and see me now?
I do see, though, you will have to play it somehow. You'll have to play it as a presenter, because currently we see just, I guess, PowerPoint as your view.
Oh, you don't see me as well?
No, I mean, can you start presentation?
Yes, I will. Yes. Well, hello everybody, and sorry for the technical problems, which all at my end, I'm afraid. And thank you very much to Joanna for standing in there when I couldn't get online. Oops, I'm not on the correct page there. Just about breeding time, I think. Probably, Chris, is what I would title it. I'm going to tell you about the work I've been doing with trees and the work I've been overlapping with John Woolliams. John Woolliams, I beg your pardon.
Sorry to interrupt you, Steve. We can't see the presentation mode of your presentation. That's what Matea was saying. So we see basically the PowerPoint view, but not the presentation mode.
Oh, okay. What do I do about that then? Where's stop sharing?
Can you just go into slideshow and then present? It may show up automatically. You see on top in the PowerPoint.
Stop sharing. Oh. Right. Let's. So you're not seeing. Are you seeing anything? No.
No.
Can you share again? And then just share again.
Yes.
And
all
then
right.
go in the PowerPoint to slideshow on top. And then just click on present. And if it doesn't work, don't worry, we can see the slides as well. We just don't see it in presentation mode.
Oh, I see.
Or I can share the presentation from my desktop if this is better, and Steve just has to tell me to go ahead with the slides.
Yeah, it might have changed a bit.
I think it's okay. We could see
Okay.
it okay. Right? Let's just move how it was. It was perfect. It's okay, Steve.
So, are you seeing the screen saying about breeding time? You're seeing that, are you, at the moment?
Now you need to share again. Sorry.
I need to share again. Here. Oh dear. Share desktop. I'm unclear as to what it is you're seeing right now, if anything.
We see your PowerPoint, and we see you as a live, as a movie, as a live.
Okay. So what isn't that it then? Do we need any more?
You should be able to play it as a PowerPoint presentation. What if you click on F5? What will happen?
F5.
Yeah. Nothing happened.
No. So if I change my screens, if I go from one slide to the next, do you see that?
Please try to go to the second.
I'm on the second slide now.
No, it's still your about breeding time slide.
Oh. All right, I don't know what's going on there. Oh dear. How do I?
Steve, do you have on your desktop at home? Are you running the PowerPoint in presentation mode, or do you have what we see that just the slides with the notes underneath? Do you have it already in presentation mode?
Well, I'm looking at my screen now, and it's just a slide with the notes underneath.
If you see on top the toolbar, you see that there's slideshow. It says slideshow. So you have file, edit, slideshow. If you click on that,
view
that's
show.
the one. If you can do this one, yeah.
Yes, I've done that. Is that making any difference?
Not to us.
No.
Oh dear. What is it?
Shall I just share the screen in that case?
Yeah, I guess so.
Matthias, that's fine.
Okay. Yes.
And then Steve, you just tell me when you want to go to the next slide, okay?
Yes, right.
Okay, perfect.
Okay.
There we go.
All right. Yes. Well, sorry about that, ladies and gentlemen. I don't know what's going on there. Probably problems at my end. We've heard about dogs, sheep, cattle, all the different animals and organisms which John has been working with, and I want to take the opportunity of telling you about some of the work which he and I have been doing on trees. I'm now a retired tree breeder from forest research. I've been retired for about a couple of years, and I was tree breeding for about 30 years, and John and I overlapped for about 25 years of my career. And I was very fortunate that John has a sort of a keen interest in trees, and he's still very active with one of the community woodlands in the Peebles area, which is where we both live. We share the hometown of Peebles. And I want to tell you about some of the work he has done to bring tree breeding more familiar with quantitative genetics, and more recently more familiar with genomic selection. So the next slide, please. Oh, who's controlling the slides now? Right, okay, thank you. So I'm going to talk about Sitka spruce in Britain. Sitka spruce is called Picea sitchensis, its Latin name. It's not native to Britain. It comes from all the way from Alaska right down to California as its natural distribution. It's planted in upland areas of Great Britain where it likes very high rainfall and is quite happy with medium to low fertility soils. We've got about 700,000 hectares of Sitka spruce growing in Britain. That amounts to about 200 million cubic meters of growing stock. It's an industry which is worth about £3 billion a year and employs about 16,000 people between forestry and the primary wood utilisation industry. So next slide, please. Our objective is construction timber. As you can imagine, most of the timber we use in this country is imported from abroad, and they tend to set the standards of timber strength, and the British grown material must meet those standards. So our objective is to, for every hectare, is to grow more volume, to make sure the trees are straighter, and as a result get a lot more good quality saw logs which meet this construction grade market. Next slide. Now we actually started the Sitka spruce breeding program in 1963, and finally got around to writing in a booklet what our objective was in this booklet here in 1972. And what you see a picture thereof is a plus tree, a superior phenotype, and we selected about 2,000 of those between 1963 and the early 1990s. Next slide, please. So the next job, of course, was to get some sort of a genetic evaluation of these very large number of plus trees through progeny testing. And we very quickly learned that progeny testing is frustrating. It takes too long and it costs too much. We're dealing with a species which has a generation interval of at least 20 years. And it took us 20 years to establish the trials to test these 2,000 plus trees. We would tend to be testing about 100 plus trees each year. Each trial would be replicated over 3 sites. Each trial costs about £20,000 or more to establish. 6 trials a year, therefore, £120,000. That's a big chunk of forest research budget at the time. And the trial sites varied tremendously in location and quality. And even in the early years, our design of these trials varied quite a lot until we settled down on what we thought was a very efficient design. And then we have a problem of negatively correlated traits. Two traits we're interested in improving, growth rate and wood density, seem to be quite strongly negatively correlated. Next slide, please. So we're dealing with a species which has got a 40-year rotation length. But of course, nobody wants to wait until 40 years before you make your final selection. And in the early days, we were making our assessments as to which of the best progeny just by simple arithmetic mean across the different sites on which these progeny appeared. But even with that low-level technology, through numerous mature juvenile correlations, we were quite confident that we could select the growth rate based on just 6 years old in the field and straightness at 8 years old, but wood density was higher at 15 years old. Next slide, please. So we started our first study to look at this mixed model analysis. The first study, which involving mixed model analysis on trees in Britain, started in 1994 to 1997, and this was actually my PhD. And I was quite keen that I needed to know more about quantitative genetics, and I'd come across John at a few meetings, and he very happily, to me anyway, agreed to be my supervisor for my PhD at Roslin. And this is when I started learning for the first time about BLUP and mixed models. And we used AS-REML in the analysis of the variance components. And I was told that this was actually the first time AS-REML had been used in any analysis outside of Australia anyway. And as a result of this study, we were able to reduce the selection age for wood density down from 15 years to 6 years, which was a considerable result as far as we were concerned. Next slide, please. There was many things which came out of the PhD, but I was always pleased about this one here, which is something that John introduced me to, a phenotypic selection ellipse. I hope you can remember this. We had 125 progenies. Each progeny was made up of 30 odd individuals, and I did not have resources to collect wood samples, wood cores from each one of those individuals. I did some calculations, and I decided that I could actually manage 46. Now, I was going to randomly select 46 from 125 until John came up with this whole idea of a phenotypic selection ellipse, which is based on work done by Cameron and Thompson in 1986, where it says if you have some prior knowledge of the relationship between two traits, you can increase the errors associated with your variance components estimation by selecting those on the edge of an ellipse, and that's what we did. So we picked these 46 with the solid dots there, and rather than the ones which the open dots in the center of the graph there. Next slide, please. And the breeding of Sitka spruce, although it might seem a bit clunky in the beginning and rather slow, it has been a success story. We consider there's about 40 million hectares of Sitka spruce, 40 million plants of Sitka spruce, about 30,000 hectares are planted each year, and this is nearly all improved material now. None of it is imported from the Queen Charlotte Islands anymore. Thank you. Next slide. And these are the sort of gains which we're getting now. Depending on exactly where your improved stock comes from, it's between 21 and 29% more volume, which is commonly stated as 25% as being more or less a mid-range there. That pretty much equates to 25% more carbon absorbed as well, which, as you can imagine, is quite a big plus these days. And we're getting greater volume per hectare, more straight individual trees growing per hectare, and so we've got a considerable increase in the number of saw logs coming out of each hectare, between 40 and 100% more saw logs. So the value per hectare has increased considerably. Next slide, please. So there was a bit of a gap. John was quite pleased, I hope, with the fact that 3 papers came out of the PhD, but eventually we started working with each other again under a new EU contract under the 7th framework called Novel Tree, which nominally went from 2007 to 2011, although we managed to continue some of the work beyond that. And this was our opportunity in forest research to try and get Sitka spruce to enter the 21st century and test out the genomic selection principles in Sitka spruce. Could we make it work? Can DNA markers speed up the breeding process, increase the genetic gains, and reduce overall costs? And the EU contract was our opportunity to investigate that. Next slide, please. Now you need to know that conifer trees are different. They are certainly compared to something like dogs, I should imagine. They're undomesticated. They have very large effective population size. They have huge genomes. So some reference points here, if it's 3 gigabytes for a human genome and about half a gigabyte for poplar trees, then Norway spruce was found out to be 20 gigabytes, and we expected Sitka spruce to be something similar to that. Now we had some very large clonal tests, which myself and colleagues from Forest Research had established in 2005, where we had very large numbers of individuals from just a few pair-wise matings, or full sib families as we refer to them. And we were able to go to just one of these full sib families and successfully get DNA out of 650 trees. So these are siblings, all from the same full sib family. And this work was published in Tree Genetics and Genomes in 2017, where it's had 3,000 accesses, which I'm not sure how you get on with some of these other disciplines we've been hearing about today, but for forestry, that's a lot. Next slide, please. And we've been able to find that the predictions, phenotypic predictions for height using predicted breeding value was 0.73. And the next slide, please. And for bud burst is 0.92, which are very, very high figures. Next slide, please. And this gave us the confidence to move forward and try and do some more in this area of genomic breeding. So again, with John Woolliams as part of the team from Roslin Institute, but this time with a contract called Sitka Spruced, which is led up by Oxford University and Professor John Mackay there. There is this new contract which is ongoing at the moment, trying to see whether genomic breeding can work more. This time we've got a large number of different families, for about 50 full sibling families, and we're going to see to what degree we can find markers and whether they are restricted to just individual full sib families or to what degree they are transferable from one family to another. Now I have highlighted on there money from industry. Now we got the money from the BBSRC, we're very grateful for that, got, I think it was 8%, and the other 20% had to come from industry. Now getting money out of the forestry industry has traditionally, for research, has traditionally been very difficult. But for this one, we found it surprisingly easy, actually. They could see the potential of if this genomic selection works and we could very much reduce the generation interval, there could be scope for turning over generations and getting some very quick gains very quickly, which of course is money to the industry. The next slide, please. And I have to mention that amongst the work which I've done with John Woolliams was we produced a monograph together. This was under the novel tree contract, with a monograph which was published by colleagues who were members of the same contract from INEA in Spain, and they unfortunately, well, fortunately, managed to flip-flop the institutes which John and I are members of. So we got Steve Lee there at the Roslin Institute near Edinburgh and John Woolliams down at Forest Research. I don't know what John thought about that, but I certainly was not complaining and considered that to be some sort of a promotion, actually. So the next slide, please. And this is the last one. So that's it about breeding time. Trees and my work with John Woolliams. I must say it's been an absolute pleasure. I've learned a lot from John. he brought us into the world of quantitative genetics, and then he subsequently brought us into the world of genomic selection. So to him and to everybody else listening, thank you, and sorry for the earlier problems. Thank you very much.
Thank you, Steve. That was really interesting. Both because I've worked with John for 35 years, knew little of his work with trees, and also because I spent a couple of £1,000 on C16 timber over the last period of lockdown. Rebuilding an 18th century farmsteading. Anyway, there is a question here from Andrea, which asking, she says, I'm sorry if I missed this, but do SNP panels exist for Sitka spruce and other species?
Well, they did not when I retired a couple of years ago, and that was the objective. Although we are fortunate in that colleagues in British Columbia are doing similar work as well. And also there's another species called white spruce, Picea glauca, which is very, very close to Sitka spruce botanically, and a lot more work has been done on that. So I must say, I knew work was being done in this area when I retired 2 years ago, but now I'm a little bit out of date. It could well be that panels do exist now, but I'm sorry, I can't definitively answer that question.
Okay, thank you very much. Well, I think thank you very much, Steve. I think we better move on now because we've lost a bit of time. Sorry about the problems earlier on.
No, mine, I'm sure.
We'll now move on to the last speaker of the session, who is the man himself, John Woolliams. And you've heard a lot about his research and contributions in the past. And his title is Where From and Where To, which I assume means he'd be looking both at the past and into the future. So over to you, John.
So can you hear me and see my screen?
We can hear you, but we can't see your screen.
We can hear you, yes, but we cannot see your screen. Please share your screen.
I thought I had, I must admit. Okay. Now I've got to get out of it again because I thought I had. Right, I'm starting again. So I'm going to share screen.
Yes, please.
And then share presentation.
Please minimize our Microsoft Teams in your screen.
Yep.
And you are all set up.
I'm all set up. And I start.
Yes, please.
Okay, so, well, thank you very much, Matea, for setting me up there. And so my talk, as Chris said, is Where From and Where To, and I've subtitled it Tracking a Butterfly. Perhaps you'll see why as I go through. But I thought I'd start, a good way to start would be to do a word cloud derived from my publication titles. And it's good to see genetic there and select and inbreeding in nice big letters. But actually, my main motivation was to see how much the preoccupations of my first 10 to 15 years of research figured in the cloud. And it's not very obvious. There they are. You'll see there, copper metabolism. Which actually hasn't been mentioned today. My mentor, Gerald Wiener, wanted someone to work on genetics of trace element metabolism and also to analyze a long-term experiment. And he took a very courageous decision to employ a young man with a maths degree, no school biology, and only the promise of an MSc in statistics. And Gerald, I don't know whether you're listening, but thank you very much for giving me that opportunity. So my first paper was actually on breed differences in copper metabolism, and the breeds included the North Romsey, which some of you may be aware of as a particularly unique genetic resource in British genetic resources, which is a seaweed-eating breed. And this paper pointed quite strongly to breed differences in copper absorption amongst the breeds. And I've got the paper there, and you can note the change times. I was the very junior author, but I appear last on the list, so times have changed. And the other task that Gerald had for me was the analysis of a long-term experiment on line breeding and crossing, with lots of measured traits on life histories, on weights, conformation, fleece fertility. And you can see some of the effects of line breeding there. You can see that these are two half sibs. One's a line cross, one's 50% inbred, and you have to guess which is the inbred one. But that experiment started before I was born and ended just after I started work. So those were the first projects. And looking back at it, it's interesting to see the progress of science in, as far as today, are basically unfulfilled aspirations. Back in that project, those projects on copper metabolism, which took us 10 to 15 years of research, we were very successful in showing substantial variation in copper metabolism between and within breeds. This had actually quite very large impacts on lamb mortality in some environments. There was a G by E associated with this copper deficiency, and also that variation was linked to hemoglobin type. And so today we'd be thinking, well, and in fact I'd be quite confident, given the data that we had, that we could have actually discovered a causative mutation for this this variation. But of course back then it was the tools just weren't there. And as far as the cross inbreeding experiment, the SIBs there, we had many lines taken to 50% inbreeding and then crossed. And even at that time, having read my Faulkner, I was aware that what I'd like to do is random regressions on lines, the line inbreeding coefficients. But then the software and the hardware just wasn't there to do it, and so I ended up doing analyses using standard linear models, but not quite what I would want. An important thread of breeding, at least for me, has always been the overcoming of sex and age limiting traits. And for example, sex and age limited, the classic one is milk yield, which is only measured in mature females, or possibly even in difficult to measure traits. And we just heard about one there from Steve, which is, for example, stiffness, measuring stiffness in Sitka spruce timber. And technology has developed over the years. It started off with what we might call biochemical genetics, which was focusing on various blood types that people had discovered, which had heritability, one which was nice, but of course there were only a handful that people could measure, and so really it never got really very far. And that, when I was starting, had developed into physiological genetics, which was about getting predictors from hormones and metabolites among pathways that people considered important, and this was very much pre-genomics. You might perhaps regard this as genetics for systems biology, or part of the development of that idea, and perhaps not very different from omic eQTLs and mQTLs we look for today. But of course, it's genomics and other omics that now dominate the solutions to these problems. But copper metabolism sort of mapped onto this physiological genetics. And for me, several other bits of work flowed from these ideas. So for one example was, which was briefly mentioned, I think today, was predicting dairy merit, focusing on growth hormone pathways, which we did with some success. And this was in collaboration with Peter Lewandal at Fulham. But that technology was not really going to be competitive with the advancing wave of genomics. Continuing the dairy theme, we then had a serious, as Mike outlined so well, we had a very serious problem of declining fertility, and I spent a lot of time working with the University of Nottingham to develop milk progesterone for helping predictors of dairy fertility. Tony Flint's group up there down there in Nottingham. And one of the outcomes of this thread of work, these physiological predictors, was starting work in what I call operational genetics. And as a non-agricultural scientist, at least when I started, I still haven't milked a cow, still inexperienced in genetics, this sort of operational genetics was sparked for me by Nicholas and Smith, which for me was a landmark. And for the first time, I realized that breeding schemes can be radically changed. It hadn't been clear to me that somehow breeding schemes were almost ordained to be as they were, but Nicholas and Smith showed that you could do dramatic things to them, and that this relied on gaining insight into the importance of technologies and how you integrate technologies into the breeding system, and that the quality of the technology was vital. And that this paper also was anticipating how technology was changing. So, for example, it did consider cloning, and this was very much before pre-Dolly, well, 10 years at least. And just as importantly for me, it also became clear that there were major gaps in theory. And these opened up different pathways for me. First of all, I could actually look at the dairy juvenile predictors that we've been working on and try and work out how well they were doing. They had some merit in particular breeding systems where you had random choices. But well before Dolly, there was a lot of talk about the value of cloning, and so I started looking at the value of cloning, and contrary to popular belief with fixed resources, it turned out that cloning was not actually useful for generating gain within the nucleus. There are other ways that you can use cloning, but actually as far as generating gain, the trade-off between intensity and accuracy when you had fixed resources was not actually beneficial for generating gain. So that work and some of the other thinking about cloning with Ian Wilmot prepared Abro and Roslin for, well, the storm of publicity that followed when actually Dolly arrived. But that thinking in operational genetics also had the idea about, came up with the idea about factorial mating. And the lesson from factorial mating is that there are actually efficient and inefficient ways of converting genetic variation that we have into gain, and that you can make changes in breeding schemes that make that conversion. I have some variation here, I want to make some gain. There are good ways of doing it and there are bad ways of doing it. And I think that is an important thing to realize. It's not a simple function. And asking what these ways may be, identified what I call unified theory. And this is expressing gain and inbreeding in a common framework. Here, just which is simply just the long-term contributions and the Mendelian sampling terms. And a pointer to this in particular, well, in this little bit about here, the sigma r squared came from work by Naomi and Robin in 1990. So for me, these equations still embody some of the challenges that we struggle with today. For example, that form for the rate of inbreeding that we see there is, for me, the clearest expression that the rate of inbreeding that we have is a property of a group. It's the sum of all the contributions of the whole population. And more than that, that every breeder in that group, or as part of that group, has an individual responsibility in its management. And I think that's, again, something that is important to make people realize, or help make people realize. We need educating people in that way, which is what Joanna was talking about there. And secondly, I find this important because it's saying that actually gain comes from aligning your Mendelian sampling terms with the contributions, and that is where the gain comes from in our populations. And that this idea is best achieved in practice by optimum contribution algorithms, which is optimum as its name suggests. And I think these algorithms were put forward by several authors, and but their importance is that they are, from day one, actually trying to align your Mendelian sampling, your contributions with the Mendelian sampling terms, according to the amount of information that you have at that time. And I think the interesting thing is that this is a technology that actually maximizes gain. Maximizes gain for whatever rate of inbreeding you have now, whatever if you implement optimum contributions, you're then maximizing gain for that. And for a community which is very quick to take up technology, the uptake of optimum contributions is, I think, patchy. And I think that many breeders conflate the idea of efficiency of generating gain, which is given by optimum contribution algorithms, with what I think is a separate idea of reducing rates of inbreeding, perhaps, or reducing risks, which is often dismissed with an assumption that, well, this will lose gain. And I think there are two separate ideas, which are often conflated. But the idea of effectiveness of selection leads to the use of leads me to the use of data to steer populations in desired directions. And so we're going back to fertility and back to dairy fertility. And Mike Coffey and I spent many years with colleagues from Nottingham in arguing for the introduction of fertility EBVs into UK dairy cattle, as you heard from Mike, and finally to good effect. And part of this involved persuading breeding companies and veterinarians that the genetic variation could be reliably captured in EBVs, well enough to give opportunities to reverse the decline. And as you saw from Mike, yes, it has. And that success in fertility obviously gave a taste for chasing data and trying to make better use of variation that existed. And so, as you see here, becomes, I get to this long relationship I had with the genetics of bovine TB, which, as Simon explained, has been a costly epidemic in UK and Ireland for a long time. But thanks to Simon and to also another Simon Rolfe in the Welsh Government, and also the old Milk Development Company, Milk Development Council, we were able to move forward, and we can answer some of the questions here. Is there genetic variation in bovine TB susceptibility? Yes, there is. Is it a testing artifact? I think we've shown that that it isn't, or at least there may be theoretical risks, as Simon pointed out, but there is something there, I think, other than that. Can we produce EBVs? Yes, we can. And can we incorporate genomics? And yes, we can. And I would like to think that as a minimum, what that work has achieved is actually about awareness and openness. Firstly, awareness of the importance and opportunity that data can give if it's collected with care, and also with a little bit of knowledge about how various disciplines can use data. And that if you get that data, and the data, once it is analysed, can be an important part of helping you manage epidemics, such as the shades of COVID that we have today. And perhaps a second area of awareness is the power of well-built models, that you can demonstrate effects exist through that. And a third point is an openness of mind to hypotheses, and the use of model comparisons to test those. So I'd like to think that that work has prompted this in all aspects of disease management and also beyond. I'd like to move towards acknowledging all my collaborators in all these work, but I want to mention two more mentors. Bill Hill, who at critical times has given me a lot of support and good advice, and Keith Hammond, from whom I learned a great deal, and just for example, how to analyze very complex, multifaceted problems which had absolutely nothing to do with formulae. And also how a lot more about a better understanding for me of breeding in practice. And one thing I know that Keith and Bill would not see eye to eye on would be on the topic of animal genetic resources. But working with Keith at FAO made me commit to all sorts of answers to difficult questions to do with in situ conservation, gene banks, all these kind of ideas in producing the small populations at risk. And ultimately, this led me to a privilege of being invited to speak at the Interlaken Agreement, which is the livestock equivalent to the plant treaty, the actual meeting when that was signed. Another outcome, of course, was to also be the person that's defined breeds for the UK government. And that's something I've always warned students never to try. But mine's the definition up on the UK government website. But now directly to the collaborators and another word cloud from the publications. And I should apologize to many of you who think but word clouds are very random, and so the sizes of names, etc. There's a lot of randomness in that. However, I do feel this brings out Beatrice and Ricardo and Steve, who's sadly missed. But these three authors, co-authors, have been with me for many years, and I've always appreciated working with them. But the point is, I'm also being very fortunate with all my collaborators And as I said to Andrea, 43 years was not long enough to work through all the interesting topics that we've had with so many people. And so there are still many things to do. There's much to do with delivering genomic tools to all our managed populations. We've just heard Steve about the difficulties in forest breeding and in companion animals that Joanna was talking about there. And there are solutions to some of these challenges, but it's going to take, I think, a lot of work, different approaches, some of them sociological, some of them educational, to actually solve them. And there are the projects that we've had at Roslin, which some of them had took to what I call the final frontier for livestock, which is several hundred thousands of individuals with sequence level data. And I think we've got there, but I think it's going to be a few years. It will take a few years to explore what that final frontier actually looks like and what we do. But with that kind of level of information, if our accuracies are not one, the questions that then flow are, well, where are the flaws of our understanding and our models and our approaches? Clearly, when I say not one, I don't mind being close to one, but if it's not close to one, what are the problems? And these are important opportunities to learn from such data. And so for me personally, where to? I'm not intending to walk away from quantitative genetics just yet. And some of you may know that I'm a member of UK's Butterfly Conservation, and I don't know whether you ever watch butterflies, but if you watch a butterfly on a buddleia, which is the butterfly bush, it will sit on it and then fly off and tour the garden before coming back to rest again on the buddleia. So I have always been attracted to the unified theories and ideas, bringing together disjointed bits of theory and practice. And maybe it's my maths background that brings me to that kind of wanting to look at those questions. And so I have some jobs that I want to do on revisiting unified theory. One question, for example, that's always asked is whether this is simply a theory for the infinitesimal model, even though that model may be a good model. And my answer to that is yes and no. I think yes, it does extend beyond the infinitesimal model, but no, I don't think I've demonstrated that particularly clearly at present. In this area, I already have a paper on variance of response that I wish to publish. That was actually handwritten 25 years ago in Wageningen, and it was actually the third of a sequence which underpin the work that you heard about briefly from Piter Bijma. And time and tide has kept me away from finishing that, so I intend to finish that. But the bigger challenge for me, I think, is to incorporate linkage disequilibrium into these formulae and see how that might help us better understand some of the oddities in existing theory. And I think this is there are ways to do that, which I'll be exploring. So I have other things to do than record moths and butterflies in Peebleshire. And to this end, to finish, I'd like to thank the contributors for their kind words, as well as for all the interesting talks, and to thank those attending. And finally, I'd like to thank the organisers for this symposium, which came as a surprise to me, and and an honour, and I thank you. And in thanking you, I'd also like to congratulate them on grappling with all the new and rapidly changing IT technology. Organising a meeting and running a meeting has completely changed from what it was as recently as 12 months ago. And well done, thank you, and congratulations.
Thank you, John. That was marvellous, and I look forward to hearing your solution about linkages equilibrium, because as you'll know, with your having contacts with Conrad and with Albert, actually what the heritability is and how you estimate it in a human population, taking account of the distribution of effects and the disequilibrium between them, is a topic of hot and often relatively acrimonious debate. So I'll look forward to your contribution on that.
Yes, I might be looking at slightly different directions. What I'm particularly interested, or at least I will name a curiosity, for example, that I've always wanted to join up. I did some work with a PhD student, Laura Corbin, a while ago, which was looking at this idea of predicting effective population size historically, many generations ago, using genomics. And this, I think, was work by Ben Hayes, and also Albert himself contributed something, sort of slightly changed the direction slightly of the way that people did it as well. So. And but there's a curiosity in that, which says, well, I can get a formula if I assume that NE is linearly related to time. And then you go on and you predict NE, which is a nice curve, potentially going up and down, and you say, oh, look, I've got NE, historical NE. It's little contradictions like that somehow, and yet they're actually quite good, some of these predictions. So there's truth in there. But it doesn't kind of stem from the way that it's being laid out. And it's those kind of things that I'm hoping I might find a bit of time for.
Well, I hope you do, John. I hope you do. There's a question here from Denise, which I think you've sort of talked about already. How do you think optimal contribution theory can be improved in the light of genomic data? You suggest we replace EBVs with GBVs and pedigree-based relationships to genomic relationships, and the optimization problem is the end of the story. Where do we go now?
Yeah. So first of all, yeah, I might not have captured all the question, but the first point is that one of the benefits of optimal contributions, if you look at its historical development, was it separated out the idea of what's my best estimate of breeding value from then the selection problem. So EBVs, GBVs, yeah, whatever you think's the best estimate of your EBV goes. Then it comes to the selection, and there I think we had this. Theo kicked off the symposium with this. With what I thought was a study I greatly enjoyed doing with Theo on this, the different measures of inbreeding that genomics comes up with. And so optimum contributions is really whatever you want to put in that inbreeding control, which brings with it the definition of what you want to do with your inbreeding. What do you mean by your inbreeding? And as Theo and Beatrice pointed out, well, there are different definitions of inbreeding when you come to genomics. Now, I think, again, one of the things that I come back to these sort of unification ideas. One thing that I thought was really interesting in the work that Theo and I had done was this idea that IBD was a kind of unifying idea to drift and heterozygosity. If you were controlling the IBD, you were controlling drift, and you were controlling heterozygosity in a predictable way. So I think, but as was also pointed out to Teo's question, I think a question that was asked Teo, might have been Beatrice, but clearly people can choose what they want to optimize, but obviously you have to accept the consequences if you haven't made a good choice, because as it showed there, if you were controlling drift, you may not be controlling heterozygosity. If you were totally concerned with heterozygosity, you might not be controlling drift. And again, in some populations, such as zoo populations, really keeping close to where you were, in fact, in many conservation schemes, that is one of your objectives. And you never know when things might happen, lots of unknowns, which of course what inbreeding is about. Sorry, I've drifted along there. Did I miss the question?
Well, I think it's a question that actually is difficult to answer. So I think you've done enough on it for the time being. But so don't go away. We won't ask any more questions. But I think what we'll do now is put Smaragda on, who wants to say a few words from her perspective and those of other students. Just hold on a moment, John. I'm sure it's under control. She'll appear in a moment.
Hello, everyone. I am Smaragda and John supervised my PhD. I am deeply grateful for your support, guidance and training of the highest quality, which has hugely influenced my career and life. John, it has been an honor and a privilege being your student and working with you. On behalf of your students, thank you. Wishing you all the best in your new retired life. Happy retirement.
Thank you, Smaragda.
Thank you. So I guess that underlines the feelings of many people here. Thanking you for your contributions, John, showing that you are a rare individual who's made important contributions in a number of important areas in animal genetics and breeding. And just to underline your rare breed status and your range of contacts, I will share another slide. Here we go. You see that?
Yeah.
Oh, there you are, you see.
Just a second. This is live. You're set up now. It's live.
Okay, so there's John being awarded the Marsh Christian Award. Now that's not something to do with swamp religion, as far as I'm aware. It's the award that the Rare Breed Survival Trust award for contributions to genetic biodiversity, and I guess it shows a couple of things. Firstly, that John has a wide range of contacts, including the royal family here, Prince Charles. Prince Charles himself actually won the award a few years earlier than that. It shows that John's surname has always been impossible for anyone to get correct. Anyway, I'll stop that now if I can.
It's only you now on the screen.
Only me, okay. So you're officially retired. As you pointed out, you're not going to retire. And as I pointed out to others earlier on, in fact, today you're already double booked and supposed to be teaching MSc students this afternoon. And I think you'll be carrying on with that, as well as the, at least for a short while, as well as the other things you talked about. But we do hope you have more time to relax and devote to your hobbies, whether it's pointing walls or recording bats and micromoss. Interesting discussions we've had in the past, and which led me to buy my own bat monitor and record the pipistrelles of two types and brown long-eared bats in our barn, and of course restoring the natural border forests. So thank you, John, and a final thank you to all the speakers for their excellent contributions and all those who attended. The talks will be available online, and remaining questions I hope will be answered.